Here we see an example using the fluorescence spectra of sugar samples that we saw before. And you recall that we had 3 outlying samples, samples 7, 10, and 71. And we knew that sample 10 and 71 were different because they had a very high magnitude. But sample 7, we could not easily see that in the spectral plot. But if we look now and plot the residuals, we can see that there's one sample that stands out, which is exactly sample number 7. All the other samples have residuals of the same and smaller magnitude. So there's something different about sample 7. In this case, it means that sample 7 has a spectral part that does not exist in any of the other samples, and therefore it's different from all the other samples. Then we have to decide what the right thing is to do with that sample. One solution could be to exclude that sample from the analysis because it's different. But before we do that, we first have to detect why sample 7 is really different. Is it because it's a wrong sample? Is it not a sugar sample? Maybe it's another type of sample? Is it a sugar sample that is just extreme? And maybe we should have just included more samples of a similar type in order to be able to make an adequate model that will tell us something about the variation we expect in the future. Those are questions you have to look into when you analyze your own data. Residuals are often used in what is called an influence plot, which is a plot that can help detect outliers. It is a plot of the residual variance for each sample versus the leverage. And the leverage was the Mahalanobis distance, showing us whether a sample was extreme compared to the other ones. And when we talk about outliers, then potential outliers are samples with high residual variance or with high leverage. And what you can see in an influence plot is an outlying sample will often be lying first in the upper part of the plot, having a high residual variance, just like sample 7 in the example before. And if you then extract more components, then you will see that because sample 7 is less well described, then the following components will be used to describe that sample, because it's the only one that's not yet well described. And therefore, in the following components, we will see that that sample will have a high leverage. So, in an influence plot, we often see a movement from the upper left to the lower right for outlying samples. You have to be careful about over-interpreting the plot. The plot is always scaled so that one sample will be outermost, but that does not mean that any sample lying on the extreme is an outlier. You have to compare that to the magnitude of all the other samples. In the example shown here, there is an outlying sample, but it doesn't seem to be an extreme sample because it doesn't seem to be orders of magnitude from the major part of the data. You can develop rules of thumb telling you when a sample is an outlier, but they're usually not that helpful because you have to understand your data. In the plot here we see that sample 101 is an outlier. Well, maybe we expect that sample 101 is a little bit different from the other ones, and therefore we are not surprised by the results shown here, and therefore we cannot conclude that the sample is incorrect and should not be included. On the other hand, if we know that sample 101 is the most normal sample we could have, then we will be surprised by it being so extreme in the influence plot, and we will perhaps decide that it has to be included. Discussions like that cannot be included in rules used for detecting outliers, and therefore we generally try to avoid using those rules, but use our common sense and knowledge about the data instead. And the final slide, we want to talk about how to do PCA in practice. The first thing you do when you start a PCA analysis is to decide on the pre-processing you want to use. Centering is almost always used, but you have to think about whether you want to do scaling of your data. If your variables are on very different scales, you would normally want to scale your data initially. But on the other hand, if you have data that are measured on the same scale, then oftentimes we do not want to scale them initially, because there is a relation between the scale of different variables. Then you start by doing a PCA with a few components. This is irrespective of whether you expect to find 10, 15 or 20 components in your final model. In the beginning you want to look into the major part of the variation, and we know that PCA is defined such that the first components are the most important ones. So those are the ones you look into initially. You use the scores, the residuals and any other plot you would like to find the most extreme outliers. The most extreme outliers often have to simply be removed because they are typically erratic samples. But you also try to detect if there are more subtle outliers, and you may have to go back and consult your original data, your logbooks, etc., to determine how to handle those outliers. Once you have a good impression about what samples are different, you start to go into cross-validation and do a little bit more careful selection of the number of components. Again, you look for outliers because these may change depending on the number of components that you decide. And you go back and forth detecting outliers, detecting the number of components until you have no changes in your decisions. And after you're done with that, you can assess your model more carefully. And you can start interpreting the scores, the loadings, visualize residuals, and use the model for whatever you decide to use it for. One note with respect to the use of the unscrambler. When you do use the unscrambler for analyzing your data, unscrambler will make some decisions for you. For example, unscrambler will decide how many components to use in certain plots, for example an influence plot. Typically, unscrambler is wrong about the number of components. You have to decide the right number of components, and therefore you have to decide how to plot these things. And that means that you have to redo your plots with the number of components that you find appropriate.